Friends, in this video tutorial we'll be talking about agarose gel electrophoresis. So let me pick a color from here at the beginning. So we'll be talking here about the agarose gel electrophoresis, and we have seen the different types of electrophoresis out there, different types of gel electrophoresis. There are agarose gel electrophoresis, polyacrylamide gel electrophoresis. Now the idea of gel electrophoresis is to separate the combination of all the DNA molecules based on their length. In case of this agarose gel electrophoresis, it's usually used to separate nucleic acid, especially to separate DNA molecules. Now you have isolated a DNA molecule, let's say from a cell, by cracking that cell open, but you don't know exactly the length of those DNA and the different functionality of the DNA. Or let's say you have a large DNA segment, you break them into smaller fragments, and then you need to take each of the smaller fragments, amplify them, and to identify their lengths and all these things. So for all those stuff, for understanding the length of those DNA fragment, we need to run the agarose gel electrophoresis for that DNA. Now this process is simply separating the DNA fragment based on their length using a polymer mesh or polymer network made with agarose. Agarose is a kind of sugar. Agarose is a kind of sugar that is present, and those sugar are added with one another. That's a polysaccharide majorly. So multiple sugar units will be added with each other to give what we called a porous network. So it's kind of like a porous network like that. So if it has small pores in different regions, and what this DNA is doing is that DNA is skipping through this pore and transferred from different locations based on applying some charge, right? Now let's say we have this polar mesh network out there, and we have one positive charge region, we have the opposite part is negative, and we apply an electric field between them. So if we apply electric field, and the molecule that we're separating is also a type of charged, for like this DNA molecule, it is negatively charged. We all know DNA is negatively charged. So obviously DNA will move from negative side towards the positive electrode that is present there, or anode that is present there. So this migration is done due to applying electricity, and that is through this agarose network, and that's what we call as gel electrophoresis, agarose gel electrophoresis. Now there are several different types of electrophoresis pathways available there. This is agarose gel electrophoresis. There is another type called as a polyacrylamide gel electrophoresis, or PAGE. In that polyacrylamide gel electrophoresis, this is for proteins. Remember, this is for proteins, right? We separate proteins based on their molecular weight using this polyacrylamide gel electrophoresis, using SDS or sometimes using native process. So here we can see that we have a mixture of DNA molecules with different sizes, and we load them in a typical agarose network, and then we apply this electrode, the power source, and then due to the presence of this electric field, this DNA segment will move. So let's see the details here in the next slides. So actually I'm using this PowerPoint from Regents Biology. It's not produced and provided by me. The original content developers are Regents Biology, so every credit goes to them. I'm sharing their material for understanding and explaining to you. Now let's say we have the DNA, long DNA. We need to cleave that DNA to smaller. Sorry for the annoying sounds though. And we have those DNA. We cut those DNA into smaller fragment using different endonuclease enzymes. It could be restriction endonuclease or any other endonuclease enzymes. So after cleaving them, and why we actually compare it, why we cleave them and compare them, we do this for forensic analysis of a particular blood sample from the victim, and also to find out who is the actual murderer or all these cases. It can be used for medical diagnostics in many cases. It can be used for paternity testing. It can be used for evolutionary relationship, or what we call as the phylogram study, phylogenetic analysis, and many more processes. So how can we compare the cleaved DNA? So as you can see it here, there are different grooves, loading regions, loading wells. They look something like the structures. and we load those DNA segments here, and then we run this. So here the thing is, how do we compare DNA fragments? DNA fragments are separated by size. How do we separate DNA fragments? Using gel electrophoresis, using a medium called gelatinous medium. The gelatinous medium that we use in this case is agarose, right? Using gel electrophoresis process. And how does this gel electrophoresis work? Let's now talk about that. I've already previously talked a little bit about it, but let's say a method of separating DNA in a gelatin-like material using electrical field. So let's say this is the condition. This is the negative electrode. This is the positive electrode. Positive electrode called as anode. Negative electrode called as cathode. Now the thing here is the DNA that we added. So let's say DNA is negatively charged. We all know that. Why? Due to the presence of this phosphate group, because phosphate is negatively charged. So as we know, the DNA that we load here will be negatively charged. So it will definitely migrate towards positively charged when we add little electricity or electric field there. So let's say this is the DNA that we put here, and then we add this DNA to migrate towards the positive charge. It will migrate further, it will migrate further. Now what we can see it here is a very important pattern, that the larger DNA segments are migrating very less distance, while the smaller DNA segments will migrate the larger distance. And that's kind of true, because the network of agarose contains small pores. Now the smaller fragments of DNA definitely will migrate to larger distances, longer distances than the larger fragments, right? So as you can see, how does that help to compare the DNA fragment? The answer is, the size of the DNA fragment affects how far it travels. I've told you, small pieces travel faster, large pieces travel slower, and thus lag behind. So again, this is the smaller fragments that are going, and these are the larger fragments. Larger fragments will lag during the separation because they won't be very much able to transfer through those pores, small pores, while the small particles will move, small DNA will move. So if we look at here, this is the whole process of gel electrophoresis. What we produce a gel. Simply we add those powder of agarose into water, and then we boil it a little bit so that the water becomes crystal clear, and then we add it and add it in a tray where it can solidify usually after very 15, 20, 25 minutes it will be solidified. Once that solidifies, then we create this and during that solidification we also create these combs, also create this wells like structure. Then we load them with our DNA sample, and then we run this gel electrophoresis. Okay, so we cleave them, run them. As you can see, larger fragments will end up in the back side, shorter fragments will end up in the longest distance in the gel. So here, as you can see, running a gel, fragments of DNA separated, we add them, load them. So this is the first thing, we cut them based on the size, and then we add them. And then finally, we transfer that, we add this power source, and then the whole process works. Once that thing is done, we never know where the DNA are placed normally, because once we add them, once we add those DNA molecules, this DNA sample, we add what is called as a stain or as a dye over there. We add some dye to visualize the movement of the DNA through the gel. And the dye is in blue color, that's called bromophenol blue. So you can visualize them moving through the gel using this blue coloration. So once that thing is done, we take out this gel, but then what we need to do, we need to stain the gel to make them visible. Because remember, what we visualize here is not a perfect visualization. It's just the movement of the front of the gel. But we don't know all these different places and bands until and unless we stain them. We can stain them with different kind of dye. We can stain them with ETBr or ethidium bromide. Ethidium bromide is a very common DNA stainer. In this case, ethidium bromide is actually a UV fluorescence molecule which give us fluorescence, orange, reddish fluorescence in presence of UV radiation. So in the UV light, we can see the red bands coming in, right? So it will bind to the DNA. So it will look something like this once we expose them to the UV light. But the thing is, this ethidium bromide is toxic, it's kind of carcinogen. So that's why people are not very much using ethidium bromide nowadays. Different things are there, different chemicals are nowadays are present there, and people are using those chemicals for staining them. So ultimately the thing is, we cut this DNA, we load them, we run the gel. After the gel is done, we need to stain them to make them visualize. So now the thing is, when we apply all these things, how we apply this DNA, I mean gel electrophoresis. If you could ask me the question, when we use this gel electrophoresis, I'll tell you where not. In all the different techniques of biology, we must have used this technique. There are two important techniques in molecular biology we can't get rid of. One is this gel electrophoresis, another one is the PCR or polymerase chain reaction, because they are the foundation of molecular biology and cell biology analysis. So let's say here we use it for the DNA fingerprinting. Now DNA fingerprinting means just like our fingerprints, that they are unique for different people. They are different for different persons' DNA. For example, sections of junk DNA that are present in our DNA sections, multiple junk segments which are not coding, non-coding segments, they don't code for any protein, and they made a repeated sequence, just like repeats like CAT, 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 or GCC, GCC repeats all the time. And each person may have different number of these repeats, and this different number of these repeats, that are called single repeats or multiple repeats, whatever repeats present there. Those repeats are varying from person to person, and the presence of typical repeats are indicating for a particular fingerprint in the genome for individuals. So we can exploit this region and we can classify different individuals in different parts. So many sites on our 23 chromosomes with different repeat segments can be present. For example, let's say this is the DNA segment, and you see this is the green color, it's a repeat of CAT, CAT, CAT repeats. Now this is 3 times repeated for the individual, let's say A. Let's say for individual B, this repeat is 5 times. Let's say for individual C, the repeat is only 2 times, like that. So something happens like this. And let's say for another one, for another person, this repeat is multiple times. Let's say 5 or 6 times here. So this is individual A, this is individual B, and that thing's varying. This repeat number is varying. And by measuring this, by knowing this, we can actually tell that there are typical trademarks. I mean, these are the markers for those individuals unique to them. Now, how can we guess that? Let's say this is an allele 1. We have repeats, 3 repeats out there, 3 CAT repeats. Right? Now we break them down with endonucleases. We cut them using, say, a particular type of restriction endonuclease, only 1 or 2 type of restriction endonuclease. It gives us this different segments. So it gives us the segment, then we get these 3 different segments, and then we load them into the gel, the agarose gel. And once we load them in agarose gel, we get this band for this repeat. And we also get other bands, but we forget about those things because they are common. But this is a unique band that we get. Now let's say these are the other bands that we get, the other 2 bands. Okay. Now let's say we have the second person and allele number 2 here. So person number 2, the same allele that we can say this is a person number 2 now, that have more repeats, say 5 or 6 repeats of that same thing. As you can see, multiple repeats, and also GCC repeats, and all these different kinds of repeats, CAT repeats in multiple times are present there. So now we again do the same thing. We cut it with the restriction endonuclease, and then we get those 3 different fragments. So obviously we load them into the gel, and we also get 3 different fragments. One, we get this larger fragment in yellow because it has multiple repeats, I mean more than 3 or 4 repeats compared to the person 1, and then we get the other 2 segments. So by comparing these two different gel pattern, what we can see that these 2 individuals, they are different individuals. This DNA are not taken from a single individual, because the repeated segment, as you can see, are different, are of different length, and that is only possible, it's because of that signature sequence of repeated regions are different for those individuals. And once we see that, we can actually tell that these are the two different persons. So that's how we can fingerprint the person in different cases and tell the difference between us. Now the second thing that we can use the electrophoresis is evolutionary relationship analysis, right? So we can compare the DNA samples coming from different organisms, different individuals, let's say mammals and amphibians and all those different individuals, and to finally we can track down and can make that phylogenetic tree based on this analysis. So let's say we load them, and what we get is something like this kind of result. So by looking at it, what we can tell is something like this. Let's say this is the turtle. The first one is the turtle, second one is a snake, third one is rat, fourth one is squirrel, fifth one fruit fly. So as you can see it here by looking at these images, but why and how we can tell that? Let's say this 1 and 2, if we compare 1 and 2, you will find most of the similarity, same region, same, only one slight difference. So definitely 1 and 2 must have been originated from a single ancestor, and that's what is going on here, single ancestor. Similarly, if you look at 3 and 4, you'll find maximum similarity between 3 and 4, maximum similarity. So definitely 3 and 4 are originated from a single individual, single ancestor. Now if we compare 2 and 3, you will see a huge difference, because you can see that in 2 and 3 there are not very much similarity at all. So we can tell that 2 and 3 are more distantly related than 1 and 2 or 3 and 4. That's definitely true, because a rat and snake has a huge difference, because a snake is an reptilia and rat is a mammal or a mammalia. So if you look at finally the fifth one, the fruit fly, it does not match with any of them, because it's completely different compared with any one of this 1 to 4. And we can tell it is far distantly related and come from a different ancestor. So we can put this fruit fly completely out at the very first place, then we make this 3 and 4 out from 1 and 2, and then 1 and 2 and 3 and 4 coming from single ancestor. That's how we can create the phylogenetic tree, and that's how we can understand the evolutionary relationship between different organisms by looking at the gel electric pattern. But again, this is not that easy, though it was given in very simple way, but actually doing this thing is very difficult. I mean, using 16S RNA sample and all these things, it's kind of tedious, and we cannot do this manually on our own. There are softwares out there who, with very tough algorithm going on to actually detect what's going on and to give us the idea where it is related or not. And we nowadays we don't use this single process, single experimental purpose to analyze the evolutionary relationship. We use bioinformatics, we use we use protein study, DNA study, we use their morphological study, and all those studies compiled together to give the idea of evolutionary relationship. But this type of electrophoresis and analysis of DNA definitely acts as a very positive experimental science to make them placed in same or different group. The third use that we can do is a medical diagnostics. Now we can compare it with the normal allele or the disease allele. We can compare them between the normal and disease allele. Let's say this is the chromosome number 1 is for the normal guy. This is chromosome for the disease causing allele. This is allele 2, disease causing allele. This is allele 1, the normal allele, the normal version. Now we can actually tell whether the individual is having this allele number 2 or 1, because the allele 1 and 2 are slightly different. Let's say the allele 1 is mutated and modified, and it gives us a simple different kind of DNA segment which is causing the disease. Now let's say here, for example, the same thing has happened in case of Huntington's chorea disease. So for allele 1, if we break it down and run the gel, we will find these 3 bands, and for the allele 2 we get many number of bands. And it is telling us that the segment or the DNA sequence between allele 1 and allele 2 are not the same. They are different. That's why even if we treat them with same restriction enzymes, we end up with different number of bands, because the enzymes are cutting different sites. So the cleavage site of enzymes are different. So definitely the DNA structure is different for them. So once we see that, we can actually tell between those normal and disease segment. The use number 4, we can use it for the forensics to study the suspect and get the victim, to study victim, as well as we can take the sample from the crime scene to actually find out the suspect. So let's say here this is the crime scene sample for normal crime scene sample, then we have the sample for the victim. To exclude that out, then we have 3 different suspects and we run gel electrophoresis, and after that what we find is kind of thing like that. Now these are the 3 different suspects for us. This is the victim, so we definitely exclude that out. Victim never victimized itself. So obviously this is the sample from crime scene. So now if we compare the sample from crime